Hello friends, this lecture is on EEG recording and 1020 electrode system. As discussed in the last lecture, what is EEG? That is electroencephalogram, means electrical brain picture, or in other words we can say that it is the mapping of the electrical activity of the brain. EEG provides an image of electrical activity in the brain represented as waves of varying frequency, amplitude, and shape. It can be used to measure the brain activity that occurs during an event, like the completion of a task or presence of some stimuli, or it can be to measure the spontaneous brain activity that happens in the absence of a specific event. The typical electrical activity that occurs in association with an event, it is referred to as the event-related potential or ERP. It can determine changes in brain that may be useful in diagnosing brain disorders. Now let's have a brief view of ERP. ERP is also referred as the evoked potential. It is the response to light, sound, electrical or any other stimuli. ERPs can be the short latency ERPs that depends on the physical characteristics of the stimuli, or it can be longer latency ERPs that are influenced by the conditions of presentation of the stimuli. Normally the responses are very small. The relatively small electrical responses to specific events can be observed only if the EEG traces are averaged over a series of trials. Let's look at this figure. Here the event is a sound that is generated by a sound generator. The large background noise in the EEG wave make it impossible to detect the evoked response to the sensory stimulus from a single trial. Averaging across tens or hundreds of trials, however, removes the background EEG, leaving the event-related potential. Now, let's learn about the EEG electrodes. Electrodes are the interface between the brain skull and the electronic device. It conducts current across the interface. The most commonly used electrodes for recording EEG signals are the wet silver or silver chloride electrode, that is mainly a gel type we are using in between. It can be a dry contact, insulated electrode and non-contact electrode. Each EEG electrodes can be represented by an equivalent electrical circuit. Now look at this figure. It shows human brain, or I can say scalp layers, and their electrical model with a wet EEG electrode. The coupling between the skin and electrode can be described as a layered conductive and capacitive structure with series combinations of parallel RC elements. If you see in this equivalent circuit, EHE represents half cell potential. Further, the different layers of the head are modeled by the parallel connections of resistors and capacitors. Here, RD and CD model represent the coupling between the electrode and the scalp. If you talk about REC, it represents the impedance mainly associated with the electrode electrolyte interface, and ESE is the polarization at this interface. RU is the resistance in series modeling the internal layers. Now, this slide shows the comparison of electrical coupling of the skin electrode interface between different types of electrodes. As we have seen, for wet electrode, we are using gel in this. Next is the dry contact. In this, there is no gel component. For insulated, we have a gap and represented by a capacitor. Then we have non-contact. In that, in addition to a capacitor, we have additional uh, RC combination for a particular cloth that will also provide a kind of impedance. Now let's discuss about the recording of EEG signals. There can be two types of recording: bipolar, unipolar. Bipolar, in which both the electrodes are at active site. Unipolar, in which one electrode is active and the other is indifferent, kept at ear lobe. Now, what is the procedure? Means how the patient is made to be getting recorded. The person undergoing EEG is asked to lie down on his back, maybe on a reclining chair or on a bed. The technician will measure the head and mark the points. Maybe it is using 10-20 system or any other, but the maximum use is 10-20 electrode system, where the electrodes need to be placed. These spots are then scrubbed with a special cream that helps electrodes to get a high quality reading. The technician will put a sticky gel adhesive on about 16 to 25 electrodes, which will then attach to various spots on the scalp of the person. This you can see in this figure also. Once the test begins, the electrode sends electrical signals from the brain to the recording machine. In between the test, the technician may instruct the person to do certain things like still, close your eyes, breathe deeply, or look at events such as flashing or a picture. That is mainly done to see any kind of abnormality or disorder in the EEG pattern. On completion of the test, the technician will remove the electrodes from the scalp. Now, let's start with the 10-20 electrode system. It is an international method used to describe the location of scalp electrodes. The 10-20 refers to the fact the actual distance between adjacent electrodes are either 10% or 20% of the total front, back, or right-left distance of the skull. It is based on the relationship between the location of an electrode and the underlying area of cerebral cortex. Each site has a letter to identify the lobe. If you remember, we have discussed in last lecture about the lobes of hemisphere and a number or another letter to identify hemisphere location. The letters FP, FT, PO, PG stands for prefrontal, frontal, temporal, parietal, occipital, nasopharyngeal respectively, and C stands for the central position. Even numbers 2, 4, 6, 8 refer to right hemisphere and odd numbers 1, 3, 5, 7 refer to the left hemisphere. The Z refers to an electrode placed on the middle line. Also, you can see that the smaller the number, it will be closer the position to the midline. The A, sometimes referred to as M, it is the prominent bone process usually found just behind the outer ear. CZ and FZ are ground or common reference points for all EEG electrodes, and A1, A2 are used for contralateral referencing of all EEG electrodes, mainly the unipolar electrodes. Nation, it is a point between the forehead and nose. Inion is bump at back of skull. Now let's see how the measurements are being done. The specific landmarks are used for essentially measuring and positioning of EEG electrodes. These are found with a tape measure or maybe with a marker. The nation is the distinctly depressed area between the eyes just above the bridge of the nose, as you can see in this figure. And the inion is the crest at back of the skull, often indicated by a bump. Marks for the Z electrodes are made between these points along the midline at intervals of 10%, 20%, 20%, 20%, 20%, and 10%. The T3, C3, CZ, C4, and T4 electrodes are placed at marks made at intervals of 10%, 20%, 20%, 20%, 20%, and 10% respectively, measured across the top of the head. Skull circumference is measured just above the ears T3 and T4. It starts above the bridge of the nose, that is mainly at FP1, and just above the occipital point, that is O1. The FP1, F7, T3, T5 and O1 electrodes are placed at intervals of 5%, 10%, 10%, 10%, 10% and 5% respectively, measured above the left ear from front, that is SE1, and back, that is O1. The same procedure can be done for even numbered electrodes on the left-right side to complete the full circumference. Now, measurement methods for placement of F3, F4, P3, P4 can be done depending upon if measured back, front to back. It can be like FP1, F3, C3, P3, O1 or FP2, F4, C4, P4, O2. They can be 25% up from the front and back points. Front point means FP1, FP2 and back point means O1 and O2. Similarly, if measured side to side, that is F7, F3, FZ, F4, F8 and T5, P3, PZ, P4, T6. They can be 25% up from the side points, that are F7, F8 and T5, T6. So this was all in 1020 electrode system that is used for mainly measuring and placement of accurate positions for electrodes on the skull surface. The oscillations, mainly the oscillations are produced as we have discussed of different frequency and shape for the EEG electrodes. The oscillations that can be measured with EEG are even visible in raw, unfiltered, unprocessed data. However, the signal is always a mixture of base frequencies, which are considered to reflect certain cognitive, affective, or attentional states. When the brain is in a certain state, the frequency patterns may change. As I have told, EEG waves are generally according to their frequency, amplitude, and shape. So let's discuss about the different types of frequencies that make the combination for EEG. First is the delta wave, corresponds to the frequency range of 1 to 4 Hz. Delta waves are examined to assess the depth of sleep. The stronger the delta rhythm, the deeper will be the sleep. Increased delta power or an increased quantity of delta wave recording has also been found to be associated with increased concentration on internal working memory tasks. Next is the theta wave, corresponding to the region of 4 to 7 Hz. It is associated with a wide range of cognitive processing such as memory encoding and retrieval as well as the cognitive workload. Whenever we are confronted with difficult tasks like we have to count 100 in steps of 7, if you have to count 100 backwards in steps of 7, then these type of waves take over. It is also associated with increased fatigue levels. Next is the alpha waves that correspond to the band of 7 to 12 Hz. Whenever we close our eyes and bring ourselves into a calm state, alpha waves take over. They are also linked to inhibition and attention. Next is the beta waves that corresponds to the region of 12 to 30 Hz. They are consistent for motor regions, that means beta frequencies become stronger as we plan or execute movements of any body part. Gamma rays that corresponds to the band 30 to 50 Hz. It relates to the perception that combines different senses such as sound and light. Short-term memory matching of recognized objects and sounds. So these were the different frequency ranges. Now let's see these activities for normal EEG. Starting with the alpha rhythm. The starting point of analyzing awake EEG is the alpha activity or alpha rhythm. It corresponds to 20 to 60 millivolt maximum over posterior head regions. It is present when eyes are closed. It will be blocked by eye opening or alerting the patient. With drowsiness, alpha activity may decrease by 1 to 2 Hz. Failure of the alpha rhythm to attenuate on one side with either eye opening or mental alerting indicates an abnormality on the side that fails to attenuate. Beta activity, it is average voltage of 10 to 20 microvolts. It is predominantly over the anterior and central regions, reacts to movement or touch. Also, it is induced or enhanced by drugs, may attain amplitude over 25 microvolts. Often it is enhanced during drowsiness or when present of a skull defect. Theta activity, it occurs as a normal rhythm during drowsiness. In young children between age 4 months to 8 years, it is predominant over the fronto-central regions during drowsiness. In adults, sinusoidal theta activity can occur over the heat regions during drowsiness. Now, this is the normal EEG pattern. If you see, this is basically corresponding for alpha rhythm, or I can say alpha frequency. It is normal 10 Hz alpha rhythm. If you see, when we are opening the eyes, it is getting blocked and it is returning to normal when we are again closing the eye. Note that the faster frequency immediately on eye closure is represented as squeak. Now we will see what can be the different kind of disorders. In this mainly we will talk about the generalized seizures. The brain's nerve cells, that are neurons, communicate by firing tiny electric signals. During a seizure or convulsion, the firing pattern of these electrical signals suddenly changes. It becomes unusually intense and abnormal. A seizure can affect a small area of the brain or it can affect the entire brain. If the whole brain is involved, it is called a generalized seizure. The two most common forms of generalized seizures are grand mal seizures and the petit mal seizure. Both forms of generalized seizures cause a temporary loss of consciousness. Now let's discuss these one by one. Petit mal seizures. These seizures involve a brief lapse of awareness. The signs can include staring, eye blinking, and lip smacking. You can see the EEG pattern for absent seizures, or I can say petit mal seizures. It starts and ends abruptly, usually it is less than 30 seconds. EEG for this shows irregular slow spike wave complexes with no characteristic location. Now next is the grand mal seizure, or it can also be referred to as tonic-clonic seizure. It is caused by abnormal activity throughout the brain. Usually a grand mal seizure is caused by epilepsy. The person may lose consciousness and may fall to the ground. Body stiffens and convulses, with your arms and legs jerking rhythmically. But sometimes this type of seizure can be triggered by other health problems such as extremely low blood sugar, a high fever, or a stroke. If you see the EEG pattern for this grand mal seizure, it is divided into three phases: tonic phase, clonic phase, and postictal phase, that is mainly the phase of unconsciousness, we can say coma. Tonic is basically referring to sudden stiffness of limb, person loses balance and falls. It lasts for less than 20 seconds. It has rapid and high amplitude spikes in the EEG pattern. Clonic phase mainly represents repetitive jerks of muscles. It lasts longer than the tonic phase. The EEG pattern of this shows rhythmic spike waves matching the F of jerk. Last part of this is the state of unconsciousness, or we can say the coma. Now let's compare all the three, that is normal EEG mainly for alpha activity, petit mal seizure and grand mal seizure. You can see in alpha activity when we are opening the eyes, those are getting blocked. In petit mal seizure it is showing spike and wave activity, and grand mal seizure it is showing tonic and clonic type of variation. Now these are some references, mainly we have taken some images for electrodes from other sites. Thank you so much.